Hey, ANPers, this is Dr. Jones checking in. This is ANP1, Lecture Test 1, Video 15. 15, yay! So we left off towards the bottom middle of page 23 because we covered, we covered the non-membranous organelles in a lecture and then the membranous organelles, and now we need to talk about the nucleus. So we are here. So, section 3.3, the nucleus contains DNA and enzymes essential for controlling cellular ... which is called an ellipsis. And I added a fourth dot because it's the end of the sentence. Although, because that's the way they did when I was in school, but I don't see people adding a fourth dot to the ellipsis to indicate the end of a sentence anymore. So is this like the second space after the period? Has it disappeared? If you know, can you please put it in the comments so that I will know? Hashtag not an English major. Okay. But hashtag grammar matters. So let's talk about the nucleus. We talked about membrane flow. We need the next. All right. Nucleus. So the nucleus is usually in the center of the cell, but like in adipocytes, it can be squished off to the side. It's still the nucleus. Skeletal muscle cells are squished off to the side, the peripheral. Yeah, we'll get there. We'll come back to that. You'll need to know it, but not today. Okay, so the nucleus, nuclear envelope is the nuclear membranes. Nuclear envelope, nuclear membrane, same thing. It is a double membrane, as you can see here, with little holes in it, and the holes are called nuclear pores. Holes in the nuclear envelope, or membrane. And the nucleoli, oh, okay, that was unnecessary. Go back. Can you undo? I think we can erase though. Bonk. Oh, it doesn't erase that. Here it does. Okay, pen color. No, we're still on. No, I need to click pen. All right, I'll get it. I meant to, yeah, this way. All right, that's a P. 1 nucleus, 2 nuclei. 1 nucleus, 2 nuclei. So nucleoli is the plural, and nucleolus is the singular. General rule, exceptions to every rule, but general rule, the larger the structure, the shorter the name. The smaller the structure, the larger the name. It's like they named the big stuff first, and so they got like the simple words, and then as they started to discover the smaller structures, as they got the ability to see them, then they had to come up with more and more terms to differentiate them. So nucleus is N-U-C-L-E-U-S. It's this whole thing. The nucleolus are the little thing inside. Now the pictures in your book, there is one single central nucleolus, although there can be multiple nucleoli in each nucleus. So the cell model in lab has multiple nucleoli in each nucleus, so don't let that throw you. So nucleus, nucleolus. So nucleus has 1 L, nucleolus has 2 L's. Big difference. And of course on the lab test, if you don't spell it within a letter, you get no credit. It has to be spelled to the letter for full credit. Okay. Oh, bottom of page 23, nuclear envelope. We talked about the nucleoli. We haven't said what they do. We've said where they are. They make rRNA. Remember lowercase r, capital N, capital A, rRNA. And they assemble the ribosomes, which you may recall are 60% rRNA and 40% protein. So the rRNA is made by the nucleoli and then combined with protein to make ribosomes. So that's what the nucleoli do. So cells with more ribosomes would have more nucleoli in the nucleus. That's right. You got it. The nucleus is the control center of the cell. Next, let's talk about histones. Histones are the proteins that the DNA wraps around. So here's the term on your picture. So in a non-dividing cell, and again, these are things that we've covered in the lab lectures, and if you haven't watched those, go watch them and then come back. I have around the 1 minute mark in every video, I think, a link to the lab playlist. So find that. So the chromatin is the DNA in a non-dividing cell. And then as the cell prepares to divide at the end of interphase, that will twist up. It coils. It's just like if you take a shoelace and twist it and like keep twisting, and then it makes like a spiral. That's what it's doing. So it coils, super coils, and it coils around these proteins called histones. And it makes little bodies called nucleosomes, which is 8 histones, DNA wrapped around 8 histones, nucleosomes. And of course, some means body, so it's a body in the nucleus. Yeah, I know. And then chromatin, as we said, is the DNA in a non-dividing cell. It is loosely coiled nucleosomes. And it's condensed about 50 times more dense than DNA by itself. And then the chromosomes is next. Chromosomes, visible chromosomes, are the DNA in a dividing cell. So during prophase of mitosis, the chromatin coils, supercoils, until it's visible in a light microscope. And that's why it's called visible chromosomes, because this chromatin is not visible in a light microscope, which were the first ones they came up with. But these chromosomes are visible in a light microscope. So when they say visible, they're still microscopic, but in basic microscope, not like a scanning electron microscope or something. Chromosomes, we talked about that. The supercoiled nucleosomes, and they're condensed 10,000 times more than DNA. So a whole lot more condensed than just the chromatin, which is condensed because it is wrapped into nucleosomes. They're just loosely coiled. All right. Genetic code is the chemical language of the cell. They are recipes for proteins. Your DNA is all the recipes for all the proteins. Which begs the question, how do you make carbs and fats? Well, it's the enzymes that make the carbs and fats. So where do you get the enzymes? They're in their proteins. So the genetic code has the recipes for all the proteins, including the proteins that are the enzymes that tell you how to make carbs and fats. Okay. And then genes. The gene is the functional unit of heredity. It is the DNA recipe for sequence of amino acids to make a protein. So a gene, in English, is a recipe to make a protein. But you can actually edit them a little bit. It's just like once you get down your recipe for pancakes. You can add a couple blueberries and you got blueberry pancakes. You can add bananas, you got banana pancakes. You can add apples and cinnamon and maybe a little touch of nutmeg, and then you've got apple cinnamon pancakes. So good. Okay, so it's a gene is the recipe that makes one protein. But it can actually make a couple of different related proteins depending on how it goes through the editing process, which is neither here nor there, so we're moving on. Audible page-turning noises, which leads us to page 24. Why is the nucleus important? It stores the information to make all the proteins of the body, which I said. What do nucleoli make? Well, they make the rRNA, and they assemble the proteins, which are 60% rRNA and 40% protein, which we've said. When will the DNA be chromatin in a, you got it, non-dividing cell? When will the DNA supercoil into chromosomes? At the beginning of prophase, if you've already had that conversation in lab. If not, as the cell prepares to divide. A nice general answer. What are the building blocks of proteins? Ooh, from chapter 2. And those are your amino acids. What is the chemical language stored in DNA that codes for proteins? That's our genetic code. And what are the 4 nitrogenous bases in DNA? At the golf course. And which binds with which? Adenine binds to thymine, guanine binds to cytosine, and vice versa. And how many nitrogenous bases code for a single amino acid? And this is not review, this is new. 3. And this is called the triplet code. Triplet. I saw the other day that some identical twins had married some identical twins, and that both couples are pregnant. And so legally, their children will be cousins. Genetically, their children will be siblings, full siblings, because they have 2 sets of parents that share identical DNA. So that's fun. All right, so let's talk about DNA. Ooh, quiz, yay. Okay, you will have to do this on the test. If I make it like a real flat look, then you get that I'm like serious, right? Like that codes is serious, doesn't it? Or is it sarcastic? That's entirely possible. Okay, so the DNA is a double helix. It's a twisty ladder shape, and there are 2 sides of the ladder, and then the rungs are the complementary base pairs. Okay, so 3 sets of complementary base pairs are how much you need to code for an amino acid. The amino acids are the things that you stick together with peptide bonds to make proteins, as long as you get at least 100 of them. All right, in no way, shape, or form will I ever ask you to know which triplets code for which amino acids. I'm only allowing you to see this part of the picture as an example that, oh look, these are amino acids, and each one codes for a different amino acid. That's all I need you to know about that. That's all I've got to say about that. Okay. But the one side of the ladder is called the template strand, and the other side of the ladder is called the coding strand. So there might be a DNA test question that says you have this triplet in the template strand. What would the complementary base pair triplet be on the coding strand? And so it's basically asking what is bound to this. So if you have an A, what on the other DNA strand will bind to the A? A T. An A, T, and T. Okay, so what binds to AAA? TTT. Okay, so if you had the triplet, okay, and there might be times where I say on the test, you have this triplet. What is the complementary triplet? And you need to know off the top of your head that triplet refers to DNA. That was 3rd blink, so you know I'm serious. Okay, so if you have the coding strand triplet TTA, what is the template strand triplet? Okay, well, what binds to T? A. What binds to T? A. What binds to A? T. So I'm going to go through this whole thing with you, and we're going to do all of them together because usually about halfway to 2/3 through, I get a couple of, so we're going to keep doing that. We're going to do this again for DNA to RNA, and we're going to do it again for RNA to RNA. Okay, and you can pause and watch these again. So here we have ACA. So, and we're DNA to DNA. So the DNA to DNA rules apply. So in the DNA to DNA rules, what binds to A? T. What binds at green is not showing up on the blue. I apologize. We've done so much red. How about a dark blue? Oh, that shows much better. Okay. And then C. What binds to C? G, guanine. And what binds to adenine? Thymine. Okay. So GTT, what binds to G? C. What binds to T? A. What binds to T? A. Okay. And TAC, T, what binds to T? A. What binds to A? T. What binds to C? G. AGC, TCG, you got it. And then GGC, guanine and cytosine, only ever bind to each other. Doesn't matter if it's DNA, RNA, DNA to RNA, doesn't matter. It's just guanine and cytosine are buds, and that's just their thing, and that's the end of the story. So CCG, and then GCC binds to CG. Oh, now y'all are making fun of my G's. Like, I can feel you making fun of my G's, and that's okay, it is what it is. All right, so, and that seems like an important thing to know, that DNA is a triplet code, and it takes 3 nitrogenous bases to code for each amino acid. All right. Oh, yeah, we're going to come back to this. Yeah, we're coming back to this. This is its own thing, and it takes its own time. So thank you, as always, for tuning in. This has been A&P1, Lecture Test 1, Video 15, all about the nucleus. So the education must go on. Dr. J out.